for inviting me to talk to you guys today. And I'm excited to tell you about some of the work we've been doing, trying to understand how loss of the tumor suppressor SETD2 can promote subclonal heterogeneity in kidney cancer. And as many of you know, clear cell renal cell carcinoma has extensive intratumoral heterogeneity. You can see here at the histological level on the left, different regions of the tumor look different under the microscope. But a lot of wonderful work from the UK, the TracerX group, has done multi-region genetic profiling in different regions of the tumors and have used phylogenetic and evolutionary analyses to try to understand how these different regions of the tumors are related to each other and how the tumor evolves over time. And through this process, their group has been able to identify that there are different phenotypes of tumors, all that have extensive heterogeneity, but there are some tumors that have significant subclonal heterogeneity, and in particular, tumors with the SETD2 mutations have more extensive intratumoral heterogeneity than tumors with other driver mutations like BAP1 or VHL. And so what we've been trying to do over the last few years is try to understand how loss of SETD2 can promote this subclonal heterogeneity, because patients with SETD2 mutations tend to have worse progression-free survival than patients with other driver mutations. And we think that that might be linked to mechanisms that promote this subclonal heterogeneity. So as many of you know, SETD2 is a methyltransferase. Its primary role is thought to be in methylating histone 3 at lysine 36, specifically the trimethylation, and it's thought to be the sole enzyme that performs this function in mammalian cells, somatic mammalian cells, I should say. SETD2 can bind actively transcribing RNA Pol2, and I should say that the H3K36 trimethylation that SETD2 puts down is a mark that actively correlates with active gene transcription. And so a lot of the work focused around SETD2 is trying to understand how it may regulate transcription. And the model for a long time has been that loss of SETD2 may alter transcription in some way that promotes tumorigenesis. Although work from the Rathmel lab and a number of other labs over the years have shown that if you knock out SETD2 in cell lines, there aren't profound changes in the transcriptome. And also, it doesn't change RNA Pol2 occupancy across the genome. So we kind of are leaning towards the hypothesis now that SETD2 loss doesn't largely change genic transcription in a way that might promote transformation and or this change in heterogeneity. There has been work from a number of different groups over the last few years showing that SETD2 loss can affect splicing, and so there's a lot of work in that space as well. So
Frank, can I
yes,
can I ask you a question about that? So that seems really unexpected, right? That's a very surprising result in terms of not seeing an effect on transcriptional output when you're getting such a profound effect on chromatin. So can you, I don't know if you were directly involved with the experiments or not, but can you tell me exactly how the experiment was done? And if that could simply be, the reason I'm bringing this up is, because when you're doing your measurement, depending on how you've done this in terms of bandwidth, you inactivate MYC, for example, right? Rick Young's lab, they had to go to great lengths to identify what MYC was doing transcriptionally, because it's a global regulator of transcription, right? So all transcription being suppressed, when you're making your RNA-seq library, if your global amount of transcript in the cell just reduced by 20 to 30%, you're going to put in 30% more RNA in when you're making your library, and all the difference that existed in the MYC on versus MYC off state is now gone. But it's not because MYC is not affecting transcription. it has to do with the fact that it's affecting all transcription, right? And so when we are normalizing the denominator, when we are normalizing our transcriptional output data, is almost always we assume is constant. So if there's a global suppression in transcriptional output, the denominator is no longer constant, and you will just adjust the ratio back to 1. So I'm just asking, could this be a technical interpretation issue, or do you actually think that there's no polymerase effect at all?
Yeah, I mean, that's a really good point. So I wasn't involved in those experiments, but I know from work that Kim has done previously, they've normalized their RNA sequencing data using spiking in S2 cell Drosophila DNA, or sorry,
Chromatin, yes.
Yeah, chromatin into the experiments to get around that fact. But I know that but there are other groups that have done this too, like the Swanton lab, for example, and I'm not exactly sure how they've normalized, but that's a really excellent point. I think that that's something I actually probably don't know the answer to be honest.
And this is specific for, like, do you know what the effect is on polymerase 1, polymerase 3, like rRNA, other small RNAs?
Yes, so that's something that we're looking into right now. I know that we haven't looked at the effect of SETD2 and its effect on like link RNAs, for example, but there is some data from Scott Hake's group here at Vanderbilt that shows that there might be an increase in the amount of rRNA transcription, but we haven't looked specifically at RNA Pol 1 or 3. And the mark that SETD2 puts down on chromatin, H3K36 trimethylation, is recognized by mediators of the DNA damage response and other epigenetic regulators. For example, DNMT3B can bind H3K36 trimethylation and to mediate intragenic DNA methylation to prevent, or the thought is that it will prevent spurious transcription of introns. And also this mark is recognized by mediators of homologous recombination and DNA mismatch repair. So one possibility is that altered DNA damage response following SETD2 loss might contribute to this increase in subclonal heterogeneity. And so there are a number of different ways that you can drive genetic heterogeneity in cell populations, as we talked about epigenetic heterogeneity, and then mutagenesis, although there is not strong data suggesting that SETD2 loss might be able to correlates or promotes mutagenesis. But one of the other main drivers of genetic diversity is chromosome instability. Here showing numerical chromosome instability, meaning loss or gain of whole chromosomes, and then structural chromosome instability. You can see here with chromosomal rearrangements, and both modes of chromosome instability could provide induce somatic copy number alterations that might be sufficient to drive this heterogeneity. And chromosome instability it can occur through several different mechanisms, but one of the most well established is through altered chromosome segregation or chromosome missegregation in mitosis. So normally, as cells are entering mitosis, microtubules grow and attach to kinetochores on sister chromatids, and then segregate these sister chromatids to the presumptive daughter cells. However, when cells have chromosome instability, you can see here at the bottom right impaired chromosome segregation. Either you can have too many microtubules attached to chromosomes or too few, and then that can lead to lagging chromosomes that fail to segregate properly. And through a number of different publications that Kim's group has had over the last few years with Cheryl Walker, they've been able to identify that SETD2 loss either through one copy here in the middle panels or 2 copy losses can promote chromosome missegregation in mitosis through the generation of lagging and bridging chromosomes. And you can see the quantification at the right, that this is a very profound phenotype where upwards of 75% of the cells 3 days after SETD2 loss have these missegregation events. And so these chromosome missegregation events can arise through a couple of different ways. One is through, as I mentioned, alterations in microtubule chromosome attachments leading to these lagging chromosomes highlighted in blue. There can also be structural changes in the chromatids that result in the generation of these bridging chromosomes. And the difference here is that lagging chromosomes kind of fail to segregate properly to one side or the other and kind of hang out in the mid-zone. However, bridging chromosomes can span the distance between the two daughter cells. And work from Cheryl Walker's lab in collaboration with Kim over the last few years has shown that SETD2 may alter the microtubule dynamics, contributing to some of these lagging chromosomes through its role in methylating alpha tubulin. Sorry if the graphic is pretty small here, but Cheryl's group showed in 2016 that SETD2 can methylate alpha tubulin, a component of the cytoskeleton that's important for segregating chromosomes, and loss of SETD2 and its tubulin methylation can promote a number of defects in microtubule dynamics during mitosis. They were able to show that generate an antibody against this tubulin mark and this tubulin methylation mark, and they can see that methylated alpha tubulin localizes to the spindle poles during mitosis. And we've been able to show recently that that loss of SETD2 and tubulin methylation increases microtubule growth rates. So in this experiment, we've expressed EB3, which is a microtubule plus N binding protein fused to GFP here. And by visualizing and tracking this EB3 comets, we can quantify the growth rates of microtubules both in interphase and mitotic cells. And what we can see is when SETD2 is knocked out, we could see an increase in the EB3 comet velocity and the microtubule growth rates both in interphase and mitosis. And increased microtubule assembly rates has been shown by Linda Warderman's group and others to promote chromosome instability. So we think that at least in part, these increased microtubule growth rates might be contributing to some of the lagging chromosomes that are forming. But as I mentioned, we also see that there are formation of these bridging chromosomes, and the bridging chromosomes we were particularly interested in because they're caused by structural changes in the chromatids. And so and by structural changes, I mean that these chromosomes are rearranged, and in some cases, these structural changes can form dicentric chromosomes, and these can arise through chromosome fusions at telomeres or at sites of double-strand breaks. Alternatively, chromosomes can become tangled through catenation and then ultimately form these bridges. And these dicentric chromosomes are important because microtubules from opposite sides can attach to two places on the same chromatid and pull and ultimately stretch that chromosome out and induce a chromosome break. And so we asked whether we were seeing changes in the structure of these chromosomes in SETD2 depleted cells. And so here we isolated metaphase spreads from SETD2 MEFs here with wild type, FLOX wild type or FLOX cells. And so we can add tamoxifen to acutely deplete one copy of SETD2 in the middle panel or two copies on the far right. And then we just asked how frequently do we see changes in the organization of the chromosomes. And what we see is that either when we lose one copy or two copies of SETD2, we see that there's at about 50% of the cells generate these dicentric chromosomes. And then if we normalize to the total percentage of the percentage of dicentric chromosomes per spread, meaning normalizing to the total number of chromosomes, we see that roughly 1.5 to 2% of the chromosomes are dicentric. And then we wanted to confirm that these were indeed dicentric chromosomes. And so we stained for the histone FOXP3 variant called SEMP-A that specifies the centromere using immunofluorescence. And what we see is that normal chromosomes should have one centromere per chromatid. And so what we see is that when we knock out SETD2, we see a number of chromosomes or chromatids that have two centromeres. And I forgot to mention this earlier, but these chromosomes look a little bit different because they're mouse chromosomes and they lack the P arm, so they don't have the classic X shape that you typically see in human cells. We confirm this in another cell line, HeLa cells, where we can acutely deplete SETD2 using CRISPR. And here what we see is that we also see an increase in the formation of these dicentric chromosomes. And I wanted to mention, we chose HeLa cells specifically because these cells do not have the alpha tubulin methylation. And so we were able to kind of uncouple the tubulin methylation phenotypes in these cells. And so we think that most of the phenotypes that we're seeing would be due to the loss of H3K36 trimethylation or potentially other methylation targets out independent of tubulin. And so one of the questions we had is, after SETD2 loss, how quickly are we seeing the generation of these dicentric chromosomes? And so what we did is we added tamoxifen and then isolated metaphase spreads from these cells over time, and we found that the formation of dicentric chromosomes peaks around 3 days after SETD2 knockout, and then kind of wanes over time. But even after 2 weeks after SETD2 loss, we still see a significant increase in the presence of dicentric chromosomes. And then we also see that the formation of these dicentric chromosomes and chromosome segregation lead to a whole host of phenotypes in interphase. Both they generate micronuclei. We see a number of cells that are binucleated, cells that are bi and micronucleated and multinucleated as well. And we also see the formation of chromatin bridges in interphase. And so one of the questions we've been kind of trying to tackle for a while now is to try to understand how these dicentric chromosomes are arising. And primarily, dicentric chromosomes are thought to arise through 3 different pathways. One is through faulty double-stranded break repair leading to chromosome fusion. Another one is through shortening of your telomeres, promoting chromosome fusion at telomeres. And the third is through the formation of neocentromeres, or a new centromere at an ectopic site on a chromosome. And so one of the most well-characterized ways is through to generate these dicentrics is through telomere shortening or telomere fusion. And so that was the first thing we tested to see whether there was altered telomeres in cells lacking SETD2. And to test whether there was telomere fusion, we isolated metaphase spreads and performed FISH for the telomeric DNA. And if you have telomere fusion, you would anticipate seeing a telomeric FISH signal between the 2 centromeres, and we did not see that. And we also quantified telomere length, and we see that when we knock out SETD2, it doesn't significantly change the telomere length. The telomeres don't appear to be shorter, at least at this time point. So we think that telomere attrition or telomere fusion might not be the primary driver of dicentric chromosome formation. Then we hypothesized that there might be faulty double-stranded break repair leading to chromosome fusion or neocentromeres. And to kind of differentiate between these two possibilities, we would anticipate that if you have chromosome fusion, you would have dicentric chromosomes that have interchromosomal rearrangements, meaning 2 different chromosomes fusing together. For example, chromosome 1 fusing to chromosome 2. However, if you have neocentromeres forming, your dicentric chromosomes should consist of genetic material from the same chromosome. So it's all chromosome 1 with 2 centromeres. And so to differentiate between these two possibilities, we collaborated with Peter Lee and his postdoc Rashmi Dahia, and asked how frequently are we seeing chromosomes with interchromosomal rearrangement, which is suggestive of potentially chromosome fusion, or dicentrics without chromosomal rearrangements, which may suggest neocentromere formation. And what we saw is that the vast majority of the dicentrics fall into the purple bin, the orange bin, chromosomes without interchromosomal rearrangements, suggesting that the primary driver might not be chromosome fusion, but neocentromere formation. So to test if neocentromeres predominate in these cells, we isolated metaphase spreads and we stained for SEMP-A, the histone variant that epigenetically marks centromeres. And we also stained for this protein SEMP-B, which is a DNA binding protein that binds 17 base pair boxes at centromeric DNA. So in normal, on most chromosomes, SEMP-A and SEMP-B should co-localize because the SEMP-B boxes are enriched at centromeric loci. And so, however, if we're seeing that neocentromeres are forming, we should very frequently see SEMP-A localizing to places where SEMP-B is not present. And so we co-stained for SEMP-A and SEMP-B, or we stained for SEMP-A and performed FISH for the SEMP-B boxes, and then asked how frequently are we seeing SEMP-A localize without SEMP-B. So the panels at the bottom with where we see SEMP-A localizing without SEMP-B would be suggestive of neocentromeres, or could be suggestive of neocentromeres. And then we just asked how frequently do we see SEMP-A and SEMP-B co-localize in these dicentrics. And we see actually the vast majority of the centromeres are forming or are co-localizing with SEMP-B, meaning that they're forming at the right location, which means that it's very unlikely that they are neocentromeres and they're just centromeres where they should be. Which means that the dicentric chromosomes are not arising through any of the three normal pathways that we would have expected. And so, I mean, frankly, to be honest, we're kind of stuck there for a little while trying to figure out what exactly was going on. But one of the things we noticed while we were doing these experiments is that very frequently the dicentric chromosomes were symmetrical, meaning that the centromeres were typically in the murine cells, the MEFs, were typically at the poles. And in the human cells, we noticed that very frequently the dicentric chromosomes appeared to be largely symmetrical, suggesting that we weren't having random reorganizations. This appears to be a more organized or stereotypic process. And so one of the things we hypothesized is that it was possible that we might be forming or promoting the formation of isochromosomes, which are these kind of rare but recurrent chromosomal reorganizations that are mirror image inversions of these chromosomes. And so here's just a kind of a few examples of X chromosomes that have undergone or have become isochromosomes highlighted in red here. And what you can see based off of this kind of schematic of what these X chromosomes look like is you can see that you end up with these mirror image inversions that lead to kind of a simultaneous amplification of incorporated chromosome arms and a deletion of unincorporated arms. And what this looks like more at a kind of a cartoony type schematic is that if you end up with a break or some sort of chromosome insult, you can have a loss of, here shown, a loss of chromosome or the P chromosome arm, and which leads to an amplification of a Q arm. And so we asked whether these highly symmetrical chromosomes are indeed isochromosomes. And you can see here using G banding, we're able to see that the vast majority, around 75% of these dicentrics, are indeed isochromosomes that have mirror image symmetry. And we wanted to confirm that using an independent approach. And so here we just performed FISH for the P arms of the acrocentric chromosomes that contain the R DNA or ribosomal DNA. And typically, the chromosomes should only have the acrocentrics, the FISH staining the P arms or the short arms of these chromosomes. However, when we deleted SETD2, we saw an increase in the frequency of these P arm on both arms of the SETD2 knockout chromosomes. And you can see this a little more nicely here in the mouse embryonic fibroblast cell lines. When we perform FISH, you can see that this very clearly mirror image symmetry of these chromosomes, suggesting that isochromosomes are quite common following SETD2 loss. And so what we think is happening is that when we lose SETD2 or H3K36 trimethylation, which I'll talk more about in just a minute, we end up with the formation of a whole spectrum of isochromosomes, some that are monocentric, some that are isodicentric, and some that are isoacentric. And so if we're forming these isochromosomes, as I mentioned, you should have kind of an amplification of one chromosome arm and or a deletion of the other chromosome arm. And so that would suggest that following SETD2 loss, you should have a number of chromosomes that might have a disparity in the P to Q arm. And so to test whether this might be occurring in kidney cancer, we queried the renal PCOG data set to ask whether loss of heterozygosity or meaning loss of one copy of SETD2 or loss of both copies of SETD2 leads to an increase in the number of chromosomes with an alteration in the ratio of the P to Q arms. And indeed, that's what we see. And we also see that the tumors with SETD2 mutation have an increase in the density of the copy number variants. So one of the questions we've been kind of trying to tackle more recently is to try to understand, well, what, how does SETD2 loss or what function of SETD2 is required to suppress the formation of these isochromosomes and isodicentric chromosomes. And so recently a small molecule inhibitor of SETD2 was developed, and we can see that when we treat cells with this inhibitor, we're able to block its methyltransferase activity, and you can see the concurrent loss of H3K36 trimethylation. But one thing that was interesting we found is that when we treat cells with this methyltransferase inhibitor of SETD2, we see that while H3K36 trimethylation is gone, SETD2 is still present. And so we asked whether inhibition of SETD2 is sufficient to cause formation of dicentric chromosomes. And you can see from the middle panel here, if we knock out SETD2 in red or treat with the inhibitor in purple, we still see an increase in the formation of dicentric chromosomes, suggesting that the catalytic activity is required to suppress the formation of dicentric chromosomes. However, as I briefly mentioned before, we also see that when we knock out SETD2, there's an increase in the rate of chromosome instability, meaning that there's changes in the chromosome number. And so we asked whether inhibition of SETD2 alters the rate of chromosome instability. And so when we knock out SETD2, there's an increase in the frequency of chromosome instability. However, when we treat with the methyltransferase inhibitor, the SETD2 inhibitor, we do not see that there's an increase in the rate of chromosome instability. Now, this is just from one cell line, so we're working on trying to test this in other cell lines. But what this may suggest is that the methyltransferase activity is important for suppressing the dicentric chromosomes, but there could be a non-catalytic activity that's important for suppressing the rate of chromosome instability. And so kind of our proposed model right now is that SETD2 suppresses genomic instability through two different ways. One is through its non-catalytic activity suppressing numerical chromosome instability, and somehow through its catalytic activity is preventing the structural chromosome instability leading to these isochromosomes. And so we've been trying to understand what kind of cellular mechanisms might be important for generating these isochromosomes, because the mechanisms for generating isochromosomes are largely unknown, especially in mammalian systems. The best work for understanding how isochromosomes form is actually in fission yeast. And so we've kind of turned to that literature to kind of try to understand or identify mechanisms that might be promoting these isochromosomes. And so one of the best characterized mechanisms for generating isochromosomes is through the loss of Rad51. So typically, as cells are undergoing DNA replication, Rad51 can bind single-stranded DNA at replication forks and anneal it to its correct location and allow for DNA replication to proceed unimpeded. However, in cells lacking Rad51, this protein called Rad52 can predominate, and Rad52, through a mechanism I won't really have time to get into today, but is much more promiscuous in how it can re-anneal this stalled replication forks, and so it can anneal to it requires less homology for re-annealing, so it can anneal to nearby inverted repeats and promote essentially a U-turn in DNA replication, leading to an iso-acentric or an iso-dicentric chromosome. And so we asked, well, sorry, I should say that this model would propose that these isochromosomes are forming during DNA replication and downstream of Rad52 activity. So we tested those two possibilities. So to do that, we generated a cell line with a Degron tagged SETD2 that we can deplete by addition of this molecule called DTAG that can deplete SETD2 in roughly 1 hour. So we synchronized cells at the G1S transition using a double thymidine block, acutely depleted SETD2 by addition of DTAG or inhibited its catalytic activity through the SETD2 inhibitor, and then released into S phase and then isolated metaphase spreads from these cells. And what we see is that either depletion or inhibition of SETD2 during DNA replication led to an increase in the number of cells with dicentric or acentric chromosomes, as well as the frequency of chromosomes that are acentric or dicentric. So demonstrating that loss of SETD2 during DNA replication is sufficient to cause the formation of these gross chromosomal rearrangements. And then we asked whether these dicentrics could be arising through a decrease in Rad51 activity and an increase in Rad52 activity. So it's been previously shown by a number of different groups that loss of SETD2 decreases Rad51 activity. So we just corroborated that data here. We synchronized cells during DNA replication and stained for Rad51 and Rad52, and Rad51 and 52 form these foci in the nuclei. And so we just quantified how frequently we see, or how frequently, maybe I'll say it this way, the penetrance of Rad51 foci during DNA replication. And we can see when we deplete SETD2, there's a decrease in the number of cells with predominant Rad51 foci and a concurrent increase in the number of cells with Rad52 foci. We then depleted Rad52 using siRNA and then quantified the frequency of or the penetrance of cells with acentric or dicentric chromosomes, and we see that in control SETD2 knockout cells, we see about 50% of cells with dicentric or acentric chromosomes. We see the same frequency when we use non-targeting siRNA. However, when we deplete Rad52, we see a decrease in the formation of these dicentric or acentric chromosomes. So that suggested to us that the defects that we're seeing in the formation of these dicentric chromosomes may be at least in part dependent on Rad52 activity and the loss of Rad51 activity, but also that they're occurring during DNA replication. And so work from Charles Swanton's group and Tim Humphrey's group as well has shown that cells with SETD2 mutations or SETD2 depletion induce DNA replication stress. And so we posited that DNA replication stress might in part be contributing to the formation of these dicentric chromosomes. And we asked whether increasing the replication stress might be able to potentiate this activity and further increase the formation of dicentric chromosomes. And so here we've taken HeLa cells, either with a non-targeting guide or a SETD2 guide that we can activate with doxycycline. And we've treated these cells either control or with low dose acetylcholine, which is a DNA polymerase inhibitor. And at low doses can induce DNA replication stress and promote chromosome breaks, which you can see at the bottom right. And we just quantified the percent of chromosomes with aberrations in these different conditions, and we can see that SETD2 loss can induce more chromosomes with breaks, and we can exacerbate that through addition of acetylcholine or through another drug that causes replication stress through nucleotide depletion and hydroxyurea. And so we see that when we treat cells with these DNA replication drugs or compounds that promote DNA replication stress, there's an increase in the amount of chromosomal aberrations that we see. And you can see in some cases they're quite extreme, forming these radial chromosomes or marker chromosomes that lack centromeres. But one surprising result that we found is that if we quantify viability in these cells using colony-forming assays, we see that treatment of cells with acetylcholine or hydroxyurea to promote replication stress, the SETD2 knockout cells, despite having more chromosomal aberrations or DNA breaks, are actually more resistant to these drugs. And so one of the things we want to try to understand going forward is why these SETD2 knockout cells, despite having more chromosomal breaks and more DNA damage, are more resistant to DNA replication stress. In conclusion, we have found that loss of SETD2 or its catalytic activity leads to structural chromosome instability and potentially loss of SETD2 through a non-catalytic mechanism promotes changes in numerical chromosome instability. We found that loss of SETD2 can induce isochromosome formation during DNA replication, potentially through RAD52 and through a mechanism we don't really totally understand. SETD2 mutated cells, despite having more DNA damage and chromosomal breaks, are more resistant to DNA replication stress. And so what we're positing is that loss of SETD2 or its catalytic activity, H3K36 trimethylation, induces the formation of these isochromosomes and isodicentric chromosomes that can form these chromatin bridges during mitosis, and ultimately these chromatin bridges can break and induce micronucleation and chromathripsis and subsequent divisions. And this could promote copy number alterations that might be sufficient to induce the subclonal heterogeneity that's been described. And so, the things that we're really interested in going forward is trying to understand more about how these isochromosomes form. So we know that isochromosomes and dicentric chromosomes are prevalent in cancer and a number of genetic disorders, and they're known to drive and subclonal heterogeneity, but to date there hasn't really been a good mechanism for their to induce their generation. And so we're excited to potentially use SETD2 loss as a tool to try to understand, one, what causes the formation of these isochromosomes, and then two, what are their consequences. And then we've shown that RAD52 can promote isochromosome formation, but that's just one possibility. There's a number of other mechanisms that have been described to promote isochromosome formation in yeast and some in mammalian systems as well. So we're currently screening other pathways to try to understand other regulators of the DNA damage response and how they might promote isochromosome formation. And then also RAD52 is not able to act on its own to induce these chromosomal inversions. So we're trying to figure out what other pathways SETD2 or RAD52 might be utilizing to induce these kind of chromosomal U-turns, inducing isochromosome formation. And one of the things that we're also curious about is, are these pathways targetable? One thing we don't know is whether formation of the isochromosomes is something that the SETD2 mutant cells require to survive, or is it something that they kind of use as a alternative pathway? And so by trying to identify which mechanisms induce these isochromosome formation, we can potentially try to identify therapeutic vulnerabilities of these cells. And then lastly, trying to use, as I mentioned before, trying to use SETD2 inhibition versus loss as a way to kind of try to understand how numerical and or structural chromosome instability drives or contributes to heterogeneity is something we're really interested in. And so I'd like to thank a number of different people from my lab and other labs, both at UMC and Kim's lab here and soon to be at the NIH, I think. And I wanted to thank Peter, who's on today, who's collaborated with us on this project and actually really broke open kind of trying the mechanism for the formation of these dicentric chromosomes, and a number of our other collaborators. And that's the end of my talk today. So if anybody has questions, I'd be more than happy to answer them.
Thanks, Frank. Appreciate it. Appreciate your time. Yeah, we have time for questions. So open it up to the group. Peter?
Yeah. Hey, Frank, that was a really nice talk. So my question is, do you know if these dicentric chromosomes, are they actually stable? For example, have you tried to isolate clones that have a specific isochromosome, and if you did, could you use that for sequencing to identify the breakpoints or what's happening at that inversion area?
Yeah, yes, absolutely. So we are in the process of doing that right now. The tricky part for us has been right now to date, we've been using these MEFs and HeLa cells, which are not great for studying. They don't at baseline they have a decent amount of chromosome instability, so it's been harder to kind of pin that down. Right now we're in the process of trying to use something that's more stable genome, like DLD1s or RPE cells, to do that experiment, but that's something we're hoping to do, because one of the things that we're really interested in is trying to understand like where these U-turns might be occurring, or these sites of inversion, and then also, to your point too, I think, is to try to understand whether these chromosomes are stable, and whether one of the centromeres is being silenced, but those are questions we haven't looked at to date.
So Frank, following up on that, so basically I was going to ask you the same question in a different way. Essentially, what is the fate of a cell in which you had a chromosomal event of this nature, right? So you're seeing the initial spike in the chromosomal imbalance, but then you're seeing a reduction as you allow the cells to remain in culture. Is that because the chromosomes are being either resolved and or being cleared out from the cells, or are those cells dying? Because that is going to really inform the mechanism that you're going to pursue, right? If you did this in a P53 proficient cell line where there was a lot of chromosomal damage being monitored actively and clearance mechanisms in place, you'll probably see a completely different cell fate compared to a P53 deficient cells, where some of these rearrangements might linger on for a lot longer. And in line with that, I think one thing that I was thinking you might want to do, especially in the context of RCC, there's already arm level loss of 3P, right? So SETD2 is gone, but so are 5 other genes that could be having profound impacts on chromosome rearrangement. And so could you, for example, generate a isogenic line where you added back SETD2? So that way, the only genetic difference between the two might be SETD2 activity or no activity. And frankly, you could put in a SETD2 catalytic dead mutant in that context, and now ask what would be the effects on chromosomal rearrangements and breakpoints, etc. And then one final idea very quickly is, again, in line with what Peter was raising, is there a propensity for a specific chromosome to become dicentric over others? because there then it becomes a biological selection question in the context of, especially in the context of cancer, right? So these are some ideas that I think would be helpful to develop this entire observation.
Yeah, those are all really excellent points. So I'll try to kind of work my way back, and hopefully I'll address some of them. But so one thing we're actively looking at right now is to try to see if there is a propensity for particular chromosomes. So Peter's data show, at least in the mouse embryonic fibroblast cells, that there wasn't a propensity for any particular chromosome, but we don't know whether that's the case for human cell lines. And so that's one thing we're actively interested in. One of the other points that you raised that is really intriguing to me in the context of kidney cancer is that, you know, the TRACERx data suggests that the SETD2 mutations occur later in tumors, and they're almost always preceded by a PBRM1 mutation and obviously VHL. And so one of the things we're also interested in is whether those PBRM1 mutations might also affect this process. So those are things that we're actively interested in. And I think, you know, to one of the earlier points that you made about oh, geez, now I'm trying to remember what the one of the other things you said was
the genetic context in which you're doing the fate of the cells where the
yes. So in terms of the kind of the trajectory, so one of the things that we've seen consistently is that different cell lines have a different penetrance for this phenotype, and so we definitely think that there is some sort of epigenetic or genetic context that allows some cells to be more likely to have these isochromosomes than others. So but we don't really understand what that is yet. But one of the things that is consistently we see across cell lines is that acutely following SETD2 loss, we see this spike in the increase of these formation of these dicentric, acentric isochromosomes. And that coincides with a lot of cell death. Now, we don't know if the dicentric chromosomes are sufficient to cause the death, but we know that they occur at the same time. And as the formation of these dicentric chromosomes wane, the viability of the cells starts to increase as well. And so those might be related, they might not, we don't really know that for sure. But one thing we do know consistently is that stable deletion of SETD2. So if we let these cells go out for months or years, we see that there is still an increase in the rate of dicentric chromosomes, but it's much smaller. So it does appear that there is some drift away from this propensity for like chronic chromosome instability and forming these dicentric chromosomes, but we don't really understand like what that adaptation might be. I think it's a really fascinating question to see kind of what causes that shift or why that shift occurs. But I think it's a really fascinating question, especially in the context of kidney cancer, where, you know, these tumors are growing for, you know, decades potentially. I think it's a really interesting question. And I think one of the things that we're really interested in is like, why do you have this really acute window to see these defects? And I think also it probably underscores why we haven't seen this phenotype in the SETD2 knockout cells before, because you need to be looking at the right time with the right approaches to be able to see these things forming. So I think it's a really, now that we have this system up and going, I think it could be a really powerful way to address some of the questions that you're raising.
So one very quick follow-up. In the MEFs or in any other relatively normal immortalized cell line, would SETD2 in interface be predominantly cytosolic or nuclear?
It's predominantly nuclear, but we do see some in the cytoplasm. What Cheryl Walker's data or has proposed is that as cells are entering mitosis, the nuclear envelope breaks down, and so there is no nucleus, and so that's why SETD2 is unleashed to methylate microtubules. But we haven't really looked at very much like what.
That's exactly why I asked that question, because I was thinking in the context of the transcriptional and the chromatin effect, and I was wondering whether the reason why you see the effect on chromatin organization but not so much on transcriptional output could be because it sits in the cytosol, and only when the nuclear envelope is gone in mitosis, that's when you see the effect on the rearrangement. Anyway, we can talk more.
Yeah.
I'm sure I'll ask that question as well.
Oh, and real quick, I also, one of the other things you mentioned, we have re-expressed a catalytic dead mutant, and we see that it recapitulates some of the phenotypes that we're seeing, but I think we need to do that experiment in a more rigorous way going forward, and looking at other pathogenic mutations as well.
Thanks both. Anything else? Any other questions from people?
I have a quick question. So you propose that when you lose SETD2, you have this switch from utilizing RAD51 to RAD52. Do you know this is also generalizable across other types of repair mechanisms, like just repair of common double-strand breaks?
Yes and no. There is quite a bit of data suggesting that if RAD51 is not functional, that RAD52 can predominate or can kind of pick up the slack, so to speak. But the best data for that is actually in yeast. And there's one thing I didn't really have time to get into, but there's a very big distinction there. So in yeast, RAD52 is sufficient to displace DNA and initiate homologous recombination. However, in mammalian systems, RAD52 is unable to actually displace DNA and initiate HR. And so there needs to be other mechanisms that open DNA and allow for this recombination event to occur. And so that's one of the things that we're really interested in. It's like, well, what is it in the SETD2 knockout cells that allows for RAD52 to come in and initiate HR? So that could be R loops or transcription in somewhat other context, but that we don't really know. So those are things that we're interested in. But what's interesting is that knocking down RAD51 isn't sufficient to initiate the formation of these isochromosomes. So it's not like just when you lose RAD51, now RAD52 goes nuts and you get dicentrics everywhere. So there is something specific about like the regulation potentially of RAD51 and RAD52 in the context of SETD2, but we don't really know much beyond that. I don't know if that answers your question, but.
I think we'll wrap there.
Okay.
Thanks all.